Crop scouting is one of the most important aspects of integrated pest management. A cultivator must evaluate the pest pressure of the crop on a regular basis, but pests aren't the only subjects to take into consideration during scouting. Both biotic and abiotic problems should be noted and recorded, as well as the efficacy of preventative and treatment controls. It is important for cultivators to become familiar with as many relevant potential pests and pathogens as possible, as well as their counters for the crop they cultivate. Having this knowledge documented along with a standing operating procedure for treatment quickens the overall time it takes to reach a decision after scouting. Ideally, this familiarization should be broadened to as many cultivating staff as possible to increase the success rate of pest detection even during non-scouting activities. Depending on the specific crop and the environment in which it exists, different scouting strategies may be more or less relevant. All scouting is a balance between time and detail. Strategies that rely on more rigorous investigation per plant, or more investigations in general, will take more time, but will also have a greater chance of finding signs of pests or disease, especially when they are less obvious. Conversely, strategies that are less in-depth might allow a greater number of plants to be scouted in a given time period. Circumstances dictate whether a focus on speed or detail is more appropriate, and knowing which is best is dependent on the objectives of cultivation and the resources available. For example, let's say there's a greenhouse cultivation environment of 8 acres, with a plant density of 6 plants per square meter, arranged in micro-irrigated rows. Importantly, greenhouse historical records show that cultivators are currently treating a Tetranychus urticae 2-spot spider mite infestation with Phytocilius persimilis at several locations. For simplicity's sake, let's also say that there are no other pest pressures or treatment records because the greenhouse has only been operating for 6 months with a dedicated scouting team of 4 personnel. In this context, it may make more economical and logistical sense to employ random sampling, wherein the scouts sample a consistent number of positions per row, but randomly select each position along the row. Random sampling has many advantages. Primarily, it eliminates biases in the scout that might lead them to sample in a pattern that unhelpfully depicts the crop's health. It is also helpful for establishing a scouting routine in general, because it is uncomplicated and can easily be modified as patterns are documented both spatially and temporally. The team may notice over time that the edges of the rows more commonly become infested, or the rows closer to ingress and egress points tend to have more infestations of a certain kind, and this may inform preventative treatment strategy. However, it is important to note that consistency is key with regards to random sampling. Increasing or decreasing the number of sampled spaces with little consistency will affect the results negatively. If time becomes an issue, decreasing samples may be necessary if additional personnel cannot be trained and deployed, but this will decrease the accuracy of results. Likewise, increasing samples will increase the accuracy of results and can be mitigated by deploying more scouts in order to reduce the scouting time of each individual proportionally. Of course, this will also have increases in labor costs per hour. Fundamental procedure may also change. A team might not have been aware of or simply didn't scout for a particular pest or pathogen, but after initial detection, it becomes more important to consider it in future scouting sessions. Some pests have a tendency to exist at certain parts of the plant, like the phyllosphere or the rhizosphere, or interior parts of the flowers, while others may appear at different locations. The more specific places that need to be searched, the more involved the scouting procedure becomes, and the more time must be allotted for proper evaluation. It can be helpful to set up a grid or chart to orient scouting procedure and keep a history of pests and pathogens encountered at a regular interval. Intervals depend on the crop size, available resources, and pest pressure, but weekly intervals are common. Some organizations supply software that can streamline the recording process of sampled areas and even statistically analyze observations of pest and treatment effects, transforming rote numbers into graphs for better readability. Merging scouting, treatment, and weather data can elucidate relationships between crop health and pest or pathogen outbreaks. When utilizing biocontrol agents preventatively, it is useful to also record their presence, either on the plant or in traps, as a representation of their population for the whole area. Over time, patterns will arise that will inform population goals for biocontrols. For example, in a sample size of 100 1 by 1 square meter spaces, a rate of observation below 50% might be associated with a marked increase in certain pests' population in the crop. This was actually my experience with Amblysia swirskyii and broad mites in a Gerbera greenhouse. If the established swirskyii population was too low during the hotter months, broad mites and cyclamen mites would establish more readily. But as long as the sampled population was at or above 50%, broad mites were much less prevalent.